Great. So then, hello and very welcome to this third workshop of the X-omics workshop series this month. Thanks for joining. We already finished 2 workshops during the last week on omics data standards and on linked data. And we also received some additional ideas for future workshops. So if there is a topic you would like to learn about more, you can also suggest that via the feedback form that is sent after each workshop, or you can also contact us via email. We're happy to get any suggestions for future events. And today is about data integration showcases. And next week on Tuesday will be the final workshop of this series. 3 of the workshop participants are going to pitch their own projects, and we'll have breakout sessions to discuss each one of those projects. And I'll move on to today. So today we have one speaker, and she will be shortly introduced by Jenny van Dongen from Vrije Universiteit Amsterdam, who will chair this session. And I will now give the floor to Jenny, so enjoy the workshop today.
Yes, good afternoon everyone. Also a very warm welcome from me. So today we have invited Ayşe Demirkan to share her experiences on multi-omics data integration. Ayşe currently works as a research fellow in statistical multi-omics at the Department of Clinical and Experimental Medicine at the University of Surrey. And she has worked in the UK since 2019, but before this she did her PhD at the Erasmus Medical Center in Rotterdam, followed by postdocs in Leiden, Rotterdam and Groningen. And Ayşe actually started her career in a clinical genetics lab. And after working in the lab, she moved towards quantitative research in the field of human genetic epidemiology. In particular, omics data integration. So her work so far has very much focused on identifying relationships between different omics layers. And she has worked with genetic and epigenetic variation, gene expression, proteomics, metabolomics, and most recently, the microbiome. In 2015, she received the prestigious Fanny Award. And currently she is leading a multi-centered meta-analysis to understand the influence of the microbiome on the host methylome. But before this, she already contributed to many national and international consortia. And today she will especially tell you about her work within the BBMRI-NL consortium, which is a very large collaboration between Dutch biobanks that was already initiated before the current X-omics initiative. So we thank Ayşe very much for being here today. Before handing over to Ayşe, I just wanted to remind you one more time that you can post questions at any time during Ayşe's talk in the chat. We will collect all your questions and Ayşe will answer them after her talk. Okay, then I now hand over to Ayşe.
So, I'm now sharing my screen, as you can see it, right? You can see the screen, right?
Yes.
Okay. So, well, thank you very much for this nice introduction, Jenny, and thank you very much for inviting me to talk here, to talk about our work in BBMRI. So, as Jenny said, I'm from the University of Surrey, but I still work with the Medical Center Groningen in the same time. So I'm going to talk about these 2 manuscripts today that were actually parts of the PhD thesis of my student from Erasmus Medical Center, Yonidiyu. She defended her PhD in 2019. That's mainly her work, which I supervised together with the PIs of the studies. So genetic epidemiology, the way we know it in 2000, in the years of 2000 or 1995, was mainly focusing on diseases and genetic variants, and those were the times that we basically the technology were not there, and extra measurements were not affordable for samples, say less than 50 or 100. So we very much focused on the genetic variants in relation to the disease. But now in 2020, the picture became much more diverse, beautiful in a sense, complex, and helps us to make a story on how a genetic variation actually affects the disease. So how is this possible? It's possible by, first of all, high density genome genotyping arrays coupled with high density imputation panels and imputation methodology. It is possible by new techniques on proteomics from plasma or from tissues. And again, new techniques on metabolome, targeted and untargeted, which can measure more than 1,000 metabolites from plasma or other body fluids. And of course, by the help of sequencing, we can do RNA sequencing, and then we can measure the transcriptome. And again, by the help of next generation sequencing or DNA barcoding, we have information of microbiome as well. And finally, now for all cohorts, we know that it is very important to collect as much as more data through online questionnaires. So we also have more information on the exposome, how these people are leading their lives, what are the risk factors in their lives other than their genetics. So it's important to understand that these data come together, but when you want to analyze it, actually we have some challenges. When we want to analyze all this data together, it is quite difficult to find cohorts that, for let's say for a cohort of 1,000 people, it's impossible to find 1,000 people who all have the transcriptome, metabolome, microbiome, environment, and proteome and the genetics measured. This is really impossible. So most of the time, these are in little subsets, actually in proportion with the prices of these technologies. So nowadays, a standard genotyping array for 500 people costs like €25 per sample, right? But if you want to measure metabolome for an untargeted array, let's say, for example, for the, let's say, metabolome platform covering about 1,000 metabolites, the price per sample is about €250. So it's 10 times of measuring the genotypes. And I think same as proteome. It is much better for transcriptome and microbiome because these are next generation sequencing based, and their prices drop continuously, but we don't see this similar price down in metabolome and proteome. Therefore, these parts of your data will always be a little bit smaller, more NAs, and it will always, you always need to decide what part of your cohort you want to send for these rather costly experiments. So we talked about BBMRI. We are going to talk about BBMRI because both of these studies I'm going to show today, one of them is partially in BBMRI and it used the data from the BIOS data set, and the other one will be the BBMRI medication study. So I want to say a couple of words on BBMRI after what Jenny said. So this is the biobanking initiative connecting the biobanks, connecting the national biobanks of Netherlands. And it has different layers. I think it was initially consisted of 6 work packages and some on cohort integration, but a lot of work has been done on making a catalog of these cohorts and arranging how this data across the cohorts can be accessible for the researchers. On the right of the slide, you see some numbers per cohort. These are the numbers taken from the integrated omics data set. So you see that there are large cohorts like 5,000 or 6,000 people, and there are rather small cohorts, which can be only case cohorts, for example, or focusing on a very particular situation, like, I don't know, people who received a cardiac treatment, for example. There can be this kind of cohorts, or there can be cohorts who have also very authentic data sets, such as imaging. So on the right side, you see on the bottom, you see the measurements done for this integrated omics data set of BBMRI. So this includes whole genome sequencing, which was actually the GoNL project of 750 people. So from this 750 people, what researchers did was a imputation panel specific for the Dutch people. But of course, also these 750 were coming from several cohorts, and each cohort also used it for their own research studies. And another interesting part of this omics data set is the part that includes DNA methylation and the transcriptome, and this is the BIOS set. And eventually the metabolomics set, which we used in the second paper I'm going to talk today. So it will be a talk about these 2 papers, but I will make some parentheses as well to go over the tools. And therefore we made a poll which Anna made. You can indicate there if you want to go over a little bit more on the tools. If you want so, just have a look at the list of tools from the list, which are like basically LD-Hub, GTEx database, MR-based tool, FUMA, and GWAS Atlas are included here. And if you want, we can visit the web interfaces of these tools and have a bit more insight, because I don't know who has already hands-on experience with them or who never heard about them. So that'd be very, very helpful. So one thing I want to do was to go to the BBMRI website, and I wanted to show the service quickly what you can do, because I wanted to show the BBMRI catalog actually. Oh, it's here. So study catalog. Oh, just a second. Yeah. So if you go to this main BBMRI website, you can see a lot of things. There are data and what is the data and how to access it, research tools about integration, ethical issues, and the knowledge share, which is basically the publications, guidelines, movies, and education. So here, if you go to the catalog, you're going to see a list of cohorts that are the parts of BBMRI, and from there, I think what's happening here is that because we are recording, or because I am in the Zoom share screen, I cannot go to this page. That's interesting, but okay, I will go on. So this catalog basically gives you an overview of the Dutch bio and data banks, and then you can find that that collections, and you can also see what is available in that biobank. Is there transcriptome data available? Is it smoking information available? Is it BMI available? And then what you can also do, there's a possibility to ask for accessing this data. It takes a bit of paperwork, but you can access the BBMRI data to use it for your own research. Of course, you will be on agreements on data sharing, intellectual property, etc., etc. But there is a wealth of data there that you can use for integration, also individual data. And the summary statistics data are also shared in different websites, which I will go over. Yeah, so we went over this. We have the RNA sequencing data, BBMR, omics, genomics, Genome of the Netherlands, metabolomics, and the methylation data from BIOS. So most of these data are collected in a cross-sectional way, and there are no repeats. Even though it says that it is a cohort study, most of the time there is not a repeated measurement of metabolomics or RNA sequencing. It's very rare. So we have these two papers. I was wondering why I was asked to join this talk, but now I understand that I used a significant amount of the word integration in the headlines of these papers. So these papers are from the work of Ayşe Niehues, which you see on the right, and we basically supervised it. So first, I will talk about what integration is. When you look at the dictionary, integration is about bringing together different elements to make a whole. So what does this include? This includes individual data pooling, which means bringing together different populations, can also be different genotyping platforms and phenotypes measured in different way. It is possible if you do data sharing agreements with trustful cohorts. It's possible to do that, and actually it is also makes it possible to make, for instance, corrections of possible confoundings. For example, population structure. You can put genetic data together and extract principal components to use for correction of population structure for cohorts coming from different ethnic background, or for cohorts coming from isolates. Or what you can also do, you can adjust for, I don't know, all other demographics to make sure that your individual data pooling is at the end the residuals are as homogeneous as possible. Another way to do data pooling is a summary level data pooling. This is what we do in a meta-analysis. And then when we run a meta-analysis, we can look at the heterogeneity and after the meta-analysis, and we can adjust our analytical method accordingly. But it could be that meta-analysis, which is coming from summary statistics from different studies, it could be that these studies did not perform exactly the same things as your cohort has performed, as your study has performed. So it is always important to make an initial analytical plan to distribute all cohorts, and then ask them to run their analysis according to this analytical plan as much as possible. A third way is integrating omics data. So this means integrating different analyses together, like transcriptomics and metabolomics and proteomics together. And how do we do that? So these are different phenotypes, but also these data are generated as a response to different research questions. So we are putting together, how does this gene affects my disease, or is this gene is a risk factor for my disease? And then the other research question is that, is this gene changing the expression of, is this genetic variant changing the expression of the gene nearby or far away? So currently there are no standard methods to correct for this heterogeneity and confounding that can happen in these data sets, because most of the time these are very valuable sets that we gather from different resources, and we have to live with what these resources has already performed, because most of the time it's a bit difficult to rerun these analyses. But I will go through some tools and to how to make things easier, and I will also explain where things don't get easier, and this is how you have to interpret your findings. So I'm going to talk about first integrating omics data and then summary level data pooling. But then I will also talk about some tools. Oh yeah, first this paper. So this was a paper we started in 2015. Actually, I was not a part of the paper then. It was initiated by a postdoc in our department, by Elena Montero, who is the second author in this paper. So initially it was a Illumina 450 methylation array, because back then there was no EPIC array in the market at that moment. So what they did is they performed an EWAS on fasting glucose, insulin, and insulin resistance measured by HOMA. So it was initially 4,808 samples. I think this includes 2 Rotterdam study cohorts, and Twins UK, and I believe NTR. And at the moment it was submitted to Nature Communications, but it was quickly rejected because I don't remember exactly the rejection letter, but it was because there was no replication cohort at the time. So that is a problem when you do things a little bit too early than others. You can't replicate, and if the sample size is small, and if your analytics is not super solid, then you get a quick rejection. So what we did, we reanalyzed this data in 2017. First, we refined the statistical models, because the initial statistical models were adjusted by only age and sex. So we included the smoking by default, and we performed the sensitivity analysis by using BMI, which later on became the core findings of the paper, actually. And then we also added a replication cohort of 11,750 people. It's not very common to see a replication cohort much larger than the discovery cohort, but in our case it works because most of the findings were actually robust, luckily, and I think that has something to do with the initial statistical models you prefer. Because the better covariates you add in your models, the robust associations you get, and these associations then would be more easy to be replicated. And then what we also did, we added this magic word integration in the title, and then it was accepted with minor suggestions. But then this integration was not so easy. So actually, it was lots of data mining, which Yoon, my student, had to go through. This was it looks like a small paper, but it was actually a lot of work. So when talking about data mining, I want to cover a couple of really bases of what kind of data is mined, and in what occasions we can do that. So when we look at the nucleus of the cells, we have these 3 packages of information that we can capture with commercial experiments, and we can generate big data. So what are those? We have the genome, genetic variants, imputed genetic variants, and then we have these modifications on the genome, which in our case are going to be the methyl additions to the cytosines, and captured by a Illumina 450 array or EPIC array, and this is what we call epigenome. So Illumina array was about 450K, and EPIC array is, I think, about 850K epigenetic markers. So these are CPGs. Some have some methyl additions on them, and some don't. And these are actually quantitative phenotypes. And then we have the transcriptome, mostly captured by RNA sequencing. And it's really important to understand that epigenome and the transcriptome are large part regulated by genetic variants, but also by environment. So although we have this information that, okay, methylation of a gene affects its transcription, usually reduces the transcription of the gene. Unfortunately, this is not 100%. This doesn't hold 100% for all the epigenetic markers for all the genes. So there is a lot of gray area. So therefore, we can't really say that epigenome is causal for transcriptome or vice versa. So this part still remains as a observational associations. And the link in between, we call them expression quantitative trait methylations. So these are basically a correlation between a epigenetic marker and the expression of a gene, which can be from different tissues, of course. That's another thing. But in most cases, this is blood. At least I will be talking about blood mostly, but I will also talk about other tissues later on. And then we have these causal links. What are those? These are the GWASs of transcriptome and GWASs of epigenome. So basically taking transcriptome as an outcome and testing whether the genetic variants across the genome associate with levels of gene expression, which we call expression quantitative trait loci. That's how we discover them. Or it can be methylation quantitative trait loci, MEQTLs. In that case, a genome is used as a predictor of epigenome, which is the outcome in this analysis. So this is the core of the integration space. This is closely linked inside the nucleus. And then we go outside of the nucleus. Then it becomes a bit more complicated. First of all, transcriptome is tissue specific. It's very tissue specific, although we use blood because it is the most accessible tissue. We know that if we want to study a cancer transcriptome, unless it is about leukemia, it's not a good idea to use blood or something about brain. So of course, it is limited by the access to the tissue. But there is a database which I'm going to explain later. And then we have phenotypes. What these phenotypes can be? They can be a disease marker, they can be metabolites, metabolomics, or proteomics, which would be the most closely related to epigenome, transcriptome, and genome. So it is also important to understand that also these observations, these correlations between the phenotypes are basically observational, and a correlation between a phenotype and epigenetic marker is also observational, and expression between phenotype is also going to be observational and tissue dependent. So EWAS here stands for epigenome-wide association study. So how we can integrate this data? So if you want to integrate data at the observational scale that are linked with observational bridges in between, you will be always have to deal with confounding of environments, of BMI, of, I don't know, smoking, whatever you can think of. So what we do, we try to remain on the causal bridges. So linking a phenotype to epigenome, you can run an EWAS, but if you check these links between genome, then you are on the causal path. So this is very important for interpretation of by interpreting data, especially when observational links are also included. So this brings me about harvesting the best from genome-wide association studies, because all the integration is circling around this 1 or I don't know million GWAS outputs, GWAS tables that we make available or we download from X website. So how do we do this? So there are, if you think of your genome-wide association output study, a table as a pie chart, a very small fraction of these results will be significant, and you will present them as, okay, these are the genetic variants that significantly associate with my phenotype. So the chance of that is less than 5 times 10 to the minus 8, right? So I'm sorry for this pie chart, which is very disproportional. But basically, there are some SNPs that can utilize this information and help you to link these top SNPs to other phenotypes. For example, what are those? I will go over. It can be FIWAs, functional annotations, physical annotations, and Mendelian randomization. Or there are tools also using suggestive SNPs regions. These can be gene set analysis, pathway, and enrichment analysis. Or there are tools who actually utilize the whole genome, and which makes it possible to test, for example, heritability genome-wide based on SNP-based heritability, which is a different measure of heritability than twin-based or pedigree-based. Or you can look at the genetic correlation between traits, or you can partition this heritability looking at SNPs that are sitting on ENCODE marks, right? So you can say that, okay, heritability partitions, but it is valid for this tissue, that tissue, and this tissue. And finally, you can also look at genetically regulated expression through metascan or PredictScan tools. So here I want to ask a question about the pool. Is there anyone interested in looking into any of these tools? Okay, so the top one is GTEx. Well, actually, they're almost, the top one is GTEx, but the other one is also very, I will just go over all of them, I think, then all of shortly, because I think people are interested in all of them. So is it okay, Anna? If I go all over them?
Sure, I think so. So just for the attendees, it might be a bit longer then, but I think
I will make
there
it
is
short.
interest.
It's mostly about showing the interface. We can't go into detail because we have limited time, but it's just making the eye familiar and the ear familiar with the tool. So one of them is the LD Hub. This is from Broad Institute. So this works on LD score regression. And what is LD score regression? Is that you can also read it here. So if a trait is genetically influenced, then variants that take more of the genome, which would have then high LD scores, should have a greater opportunity to take causal variants and therefore have higher test statistics on average than variants that have low LD scores. It's a bit complicated way of thinking, but it works very well. So in this way, you can discriminate from genome-wide inflation. You can partition the genome-wide inflation to real results and to true from spurious results and between spurious results and true results and bias. But there are very interesting uses of these methods. Now it sounds a bit vague, but heritability estimation is one part of it. It is quite arguable because these heritabilities, SNP-based heritabilities, are always lower than twin-based heritabilities or pedigree-based heritabilities, even in the narrow sense. But it still gives an idea of what is possible. So I will go to the website. Now you see the website. This is the LD Hub. These are the papers. There's one paper. So, okay, this is the paper that explains LD Hub, but there are actually 2 or 3 seminal papers that explains the LD score regression, genetic correlation, and partitioning heritability, mostly from Matt Sullivan and Hilary Funicane. So very interesting papers. You can find the link to those papers here. how to get started with LD Hub. You sign with your Google account. So you die. If you don't have a Google account nowadays, just go die, kill yourself. So what you can do, you can go to test center. That helps you to basically, without downloading the LD score tool, which is available actually, when I would recommend you to download it and use it there. But you can also, if you want to check only one trait that you don't need a high performance computing, you're not parallelizing 1000 GWASs in the same time. So you can simply just do it here on the website itself. You basically, you upload your original file, which is from you, and you select data. I'm not going to upload a file now, that's why I can't go further. But then you select data, which is already in the database of LD Hub. And the LD Hub is going to edit your files, make sure that all these are limited to HepMap SNPs, and they're going to calculate LD score for each SNP and make intermediate summary statistics files, which are QC'd, and these summary statistics files will be used for estimating SNP-based heritability. This is one way to use it. Or what you can also do, you can ask the tool to estimate genetic correlation between your trait which you studied in your class and another trait which is in their database. So this can also be done. And what is also interesting, there's a lookup center. So in this lookup center, you can browse among genetic correlations across the traits which were already done. So I think this is 173 times 173 matrix. But here on the first column, you see the heritability and the standard error of heritability. And then here you can see the heritability results. And in the other table, which is more interesting, so this is the big matrix I'm talking about. You can see the correlation, genetic correlation between one trait and another trait. And for example, if we just browse down, what is interesting, age at menarche and BMI, for example, negatively correlated. For example, childhood obesity. So what this actually helps with, I find this very useful, because what it actually brings out is that correlations between traits that were not measured in the same population. So that makes it very interesting. Of course, this is also because these are based on genetic correlation, and it will always be limited with the model that you that these, for example, there is an anorexia nervosa and BMI. It is negatively correlated. It is significant, which is also biologically very possible, right? What this shows is that so these traits are correlated, even though they are not measured in the same population. We link them through GWAS. And what I want to say is that there is always some thing to consider here. Also, each GWAS can be coming from different models. So if you run an anorexia GWAS by adjusting by BMI, perhaps you're not going to see that correlation. So this is something to keep in mind when looking at, when comparing GWAS tables in this manner. So I think this is about LD Hub. And I'm going to talk about the next one. This is also a very interesting one, using the whole genome. So genetically regulated expression. What is this? We talked about transcriptome and EQTLs. So what they did is, in this lab of Hakim Lab, they made a tool called PredictScan. and as well as metascan. PredictScan works on individual phenotypes, whereas metascan summary statistics data. And what they did, they used GTEx consortium data. They build up elastic net models for gene expression. And then what you can do, you feed your GWAS into the PredictScan algorithm, and it tells you by pooling your effect estimates and feeding them into these models, it tells you if likelihood of or liability of an increase of your trait X or disease has also has something to do with the increase in genetically regulated expression of a gene. So this is quite useful, very interesting, a very interesting tool to use, and I like it a lot. But there are a few things to consider because the models are coming from the GTEx portal. So I will just shortly go to the GTEx portal and briefly show what this is about. So this is genetics of tissue expression portal. It started from a very small set, but now it is about, let's look at the data sets, current release. So now it is about almost 1,000 donors. So who are these donors? The donors are the spouses of people who passed away. So these are, of course, to make such a study, you need to collect donors material to be able to collect this wide range of tissues. So here you can see that there are 54 tissues, 94 of them has eQTL analysis, and number of samples are around this. So this should be, I think, number of tissue samples. And what you can see here is a number of tissues, number of samples per tissue. You can see that some tissues going up to 584, for example, tibial artery, and adipose tissue is 581, can be as small as kidney cord is only 73 people. So this is really important to see that if you are looking at genes in kidney cortex in your PredictScan models, you are dealing with a algorithm with an elastic net model of coming from only 73 samples. So this is, or 73 genotypes, let's say, so 73 unique individuals. So you can understand that already that the comparisons of these methods of genetically regulated tissue expression wouldn't be fair. So you can't really say that, okay, this is not expressed in kidney tissue, but it is expressed, this associated with the expression in the muscle tissue, because then you would always have a possibility of false negativity when you look at the genes expressed in kidney cortex. So this is something to take in mind. But other than this, if you have a good sample size in the tissue model, this is a very, very interesting website. For example, you are looking at a GWAS of glucose. You can basically specify your tissue to pancreas, and then you can reduce your multiple testing correction, or you can specify your expression results to the adipose tissue, and you can even narrow down the genes of interest to a pathway if you wish to. So that helps a lot with what linking our GWAS phenotype to a expression in a certain tissue. So it really makes nice stories. So what I wanted to share, so here another thing is that because these are donors of people who passed away, female ratio is extremely low. So in most epidemiological studies, we have more females contributing recruiting than males, and here is also the case. So when a male passes away, their suppose is more likely to donor the tissue than when the female passes away, or more likely to recruit in the study, let's say. And of course, again, it's mostly white, but there are also other ethnicities. So this is about GTEx and PredictScan. So I wonder if there are already questions. No? So I will also shortly talk about FIVAs. I'm looking for my mouse. Yeah. I will shortly talk about FIVAs. So
Yeah.
yes?
Briefly interrupt. Sorry, there was one question.
Yeah.
Would you like to answer it now, and instead of waiting until
let's have a look.
We have one question from Peter Bram. Maybe we can unmute him so that he can ask the question.
Always very welcome, Peter Bram.
So this was a question about how you can determine the causal relationship with data from methylation QTLs and eQTL. So this was on your slide where you had this nice graph with all the relationships between the different omics layers.
Yeah. Yeah, it is very well. You could do Mendelian randomization, of course, but I think that really depends on as in all Mendelian randomization, that really depends on the instrumental variables. Hi, Peter Bram. You are on mute.
Yeah, great. Good to hear you talking. And well, I think the question goes back already quite a few slides, so I'm not sure that we should really deal with this now. But yeah, I think for the audience, it was important to understand what your definition of causal and observational is. And I think the use of genetic instruments as a principle is probably very important in your talk. Yeah, I would appreciate if you elaborate a little bit on that when you come across that in later on in your talk.
Yes, sure. Yeah, sure. I think, yeah, I can already say something. Causal, I will get back to that. Causal, it doesn't mean, first of all, it doesn't mean direct, right? So there can always be an intermediate phenotype. So what do you want me to can you be more specific, Peter Bram?
I think we can deal with this later in your talk. You just go on right now, and then things will become more clear, I think.
Okay, thank you. So I will just shortly go over the GWAS Atlas, which was also mentioned and pooling the participants. So I tried several FIVAS databases, honestly. And I have to say that the best one is the GWAS Atlas from Kyoko, from Daniele Postuma group. So first of all, what you can do, you can browse off a lot of GWASs. What is different than the GWAS catalog is that you get direct access to the full summary statistics, and then you can compare GWASs, and you can immediately perform a FIVAS study like super quick. But the thing is, for example, let's make one now. My favorite SNP. So this basically what it does is that there are 4,155 GWASs in this catalog of GWAS, and immediately checks the p-values for this SNP in all traits. So what you can do, you can sort them alphabetically p-value. I like domain and p-value, and you will see that there are different domains which are shown on the right of the screen. So there are a lot of traits here from UK Biobank. This is a bit of the problem. So you may want to refine your traits early on. For instance, activities domain includes very interesting questions, and some environmental questions are really interesting. So it could be, the domains could be curated in a different way. So you really need to look into what this exactly trait is. But it is good for quick lookups. And then if you want to perform, for example, a more sophisticated analysis, like looking for real co-localization, you can download these GWASs from the GWAS Atlas database, and you can perform the analysis you want to perform, further analysis you want to perform. You can also enter gene names here. So what else is here is that, well, so that's the browse GWAS function. You can search for any GWAS, and then it will take you immediately to the GWAS results. You can see PMID, where this GWAS was downloaded, Manhattan plot, QQ plot. This is supposed to be a gene-based plot, but no genes were significant, so it performs gene-based analysis using Magma. And also from here, there is some also extracts the leading SNPs. And the sister, a sister website is FUMA. So FUMA, you can upload. I will not go there, but what you can do, you can upload your summary statistics data, and you can get functional annotations rather quickly. So what else do we have? And finally, we have the MR base. I wonder if this was in the pool, but I'm constantly looking for my mouse. Yeah, so there is this MR based tool as well. I will always recommend to download this tool on your computer and run it yourself. But it is also possible to make a quick lookup. Of course, you need to have a Google account, and then get started. Okay, so you can basically perform analysis either by manual upload, or you can select some data sets which are already in catalog. So and then can be gene expression QTLs or metabolite or methylation levels, and you can use these data sets to perform a Mendelian randomization. So coming to this question of how can you perform how can you show causal association between gene methylation and expression, gene expression? So as I said, this correlation, I mean, we see correlation between them. Yes, that could potentially be a correlation that is based on a genetic variant, or it could be that the gene expression is really being affected by the methylation, but this is shown that this was not always true. So what you could do, you could perform Mendelian randomization analysis, but then you would have a big problem identifying correct instrumental variables for your Mendelian randomization, because both the expression of the gene X and the methylation of the gene X will possibly go to the same genetic variant, and therefore it would be difficult to find genetic variants that only affects the expression and not the methylation, or vice versa. I didn't do it, but this is what I think. Okay, so that was a bit on the Mendelian randomization tool. So what we did in this study, what we did in this study is that, so we had this picture. What was tested by us was number 4. So we associated the methylation to the glycemic traits. And then we tried to fill in this number 1, 2, 3, 5, and 6 by integrating data. And that was the part about data mining and data integration. For parts 1, 2, and 3, we downloaded the results from the BIOS QTL browser, which I already mentioned in the BBMRI. So one problem here, it's not only here, but in several tools, it is also a problem in the GWAS Atlas from a different way. One problem is that this BIOS QTL browser, when they did this analysis, of course, they didn't store the genome-wide association study statistics for 450K methylation locus, right? Or for all the RNA expression profiles. What they did is they performed this on the fly, and they only stored the results which has FDR less than 0.05. Therefore, a problem is when you insert a SNP here, you can't know if your SNP is not found or it was not found because it was not associated with the trait with this FDR. So I think most of the SNPs we found that was okay, but for a couple of SNPs we had to check proxies, which was quite painful. So and number 4 is our own results, so that was shared in the Figshare. And then number 5, what was that? That was linking the genetic variant to the glycemic trait. And that was, we had downloaded from the MAGIC investigators. So that was the GWAS results of glucose. And finally, how we linked the gene expression to the glycemic trace was through performing predict scan and metascan on the GWAS of glycemic traits on the models of gene expression provided by GTEx. So that was the part I already talked about. So as I said, what one needs to be careful here is that some SNPs, you can't really know if SNPs are not tested or they are not significant, although we found it a very useful website. Otherwise, we wouldn't be able to perform this analysis. So thank you, BIOS QTL browser, always very nice. And another problem was that sample size, sample size, and therefore power across this analysis is always uncomparable. So for a GWAS, you have way more sample size than for number 4. In our case, it was 4,000. And then you would have the smallest sample size if you go to the tissue-specific gene expression. So coming back to here, this integration place. So these are the parts that I think one needs to be careful about. So you always have a unfair comparison of because of unfair power across the studies, and causal links are not direct. This is really something we need to keep in mind, because there may be always mediation which we may not be testing. And this methylation and expression are very tissue specific, and in all the data sets there are different adjustments. So one needs to look at the adjustments of the data sets, how the analyses were performed in that study. And another problem is SNP imputation. I will get back to that. And if you are using positions, the genome build is another thing you would like to consider. So I will talk about the other projects, which is about summary level pooling. But did we already finish 45 minutes, or how are we doing time-wise? So
currently 3 o'clock.
Yeah, it's been already 1 hour.
Yes.
Okay. What should I do then? Should I talk about this metabolomics medication project, or should I go talk about a little shortly about the platform we are building on to ease data integration? What do you think, Anna and Jenny and Jessie?
Yeah, whatever you think is best for you. So, and I think we are still broadcasting until 4, right? So we do still have time.
Okay, then I will keep that summer level data pooling short, because the take-home messages are quite clear. That was a meta-analysis study which was performed by Dream again. So this is BBMRI. So BBMRI funded this metabolomics platform measurements for lots of cohorts, more than 10 for sure. I think it was initially it was about 25,000 persons. So we all sent plasma to the Nightingale platform for them to measure these 150 metabolites. Actually, they are not metabolites. These are macromolecules, mostly lipoproteins, fractions of lipoproteins. And from these 25,000 people, several cohorts, we recruited the ones which had medical information recorded at the time of blood drawn. And this medical information, medicine information, was then converted to ATC codes. It was more than 200 codes. I don't remember how many. There were several, too many ATC codes, unique ATC codes. And what we did, we truncated those ATC codes to ATC drug categories. So by this way, for instance, all the statins, all the lipophilic statins were merged under the name lipophilic statins. All the hypertensive drugs were merged under their categories. So a pharmacist, Lies Lewis, helped with doing this. So that was basically that was a meta-analysis, but it was a difficult meta-analysis. And I will just shortly go over the study. So we start with the meta-analyzing each medication category with each metabolite coming from each cohort. And this was all medications. So we had initially 2,000 significant associations after correction for multiple testing, and then we included BMI and smoking in these models, and then we still have 1,600 significant association. So we said, okay, this is possibly due to the confounding of medications with each other, because there were people who were using multiple medications that could have confounded the results. So therefore, we performed additional sets of analysis. What we first did is that first we looked at a subset of 7,000 people from all cohorts. These people were using only one medication. So we use it as a sensitivity analysis. And in parallel, we looked at the correlation from between all medication categories, and we identified the medications that were prescribed together, and these medications were included as covariates in the respective models. And then we still have 1,000 significant associations. And then next, we checked for the effect of the disease, because it could be that your metabolites were high because of your disease, because you have diabetes already, not because you used metformin afterwards. So to do that, we went for three things. One was to perform Mendelian randomization from disease to metabolites. Just a second. Yeah, the first option was to to perform Mendelian randomization from diseases to metabolites to check if the diseases actually has any causal effect on the metabolites. So for this one, we used hypertension, type 2 diabetes, and major depression as diseases. And second, what we did, we associated these drug-related metabolites with the disease in individuals who were not receiving a treatment, and that was the second phase. And finally, we performed Mendelian randomization from medication to metabolites, and that was only possible for statins. And when we were doing this, what we did, we used the genome-wide association study of this platform, which we had published earlier. And what we also had to was that for each of these analyses, we had to use a different subset of the studies. Because why? From various reasons. Most of the times that was because cohorts X and Y didn't have manpower to perform this analysis. So we did the analysis only in Radboudumc study and NEO, for example. And sometimes we had to use individual data for some of this analysis, and then we could only use Erasmus Rückven family and Radboudumc study, of which we had access to the individual data from. And finally, for major depression, luckily there was a paper running in parallel by Mariska Bot, and this is what we used for testing these effects for depression. So eventually, we ended up with these 15 categories, which were positively or negatively associated and significantly associated across the metabolites, which were mainly lipid fractions. So what is interesting here is that, well, this is the nature of the panel, is that there is so much redundancy. So there is a large block of VLDLs, for example, that associate with the outer layer beta blocking agents. And so overall, you see the clusters. That's because that's the nature of the panel, because many of these fractions are highly correlated for each other. So eventually, we ended up with this set, which we thought were not confounded by co-medication and the disease status. And all the results from this study were put on a BBMRI website, and it is searchable either by medication or by metabolites. So we had a lot of challenges in this study. I am happy I forgot most of them now, but I think one of the biggest challenges was the organization, organizing a large study, and dealing with the heterogeneity among the cohorts. So what I also showed in the beginning in the BBMRI website is some of these cohorts were only consisted of people with I don't know, heart disease, or people who went under surgery. There were very special cohorts, and we had to exclude them in certain parts of the analysis to have a homogeneous sets, to have a better assumption. So data cleaning was a big part of it. Getting the data from the cohort was a big part of it. Some people just were not so collaborative. And another problem was dealing with cohorts who had no analysts with computing experience. So we also had to, well, Yoon had to analyze some of the cohorts herself, and obviously there were too many significant findings, and it was a bit difficult to narrow it and find the highlights. So we had to show only the showcases, and we couldn't actually interpret all the results. And another thing I need to say is that I think for me the biggest challenge in interpretation of this data was the varying power in each analysis, and that for different layers of analysis we had to assume that these 2 or 3 cohorts which were included the analysis were actually representing the whole population. For example, the co-medication metrics were coming from the cohorts of Rotterdam and Lifelines Deep. And we had to assume that they were representing the whole medication, co-medication structure in the rest of the cohort, and which may not be always true. But well, we had to live with it, and that was the best we could do. And I hope in the future these things will be more easier. But obviously, we need more manpower. That would make it easier. I will shortly talk about this platform that we're developing at the moment. So this is a platform to make data integration easier. This is something we are developing for our work at the University of Surrey together with Inga Prokopenko. So one biggest problem for us is always, at least for me, when integrating data, is that some SNPs are just not found in some data sets because imputation platform, because QC, because other things. And this is a big problem, because then we have to go for proxies, and finding proxies and using proxies, it is difficult to standardize, and it's not always reproducible. So there are some tools which makes life easier here. For example, one tool we use is SSIMP. The whole idea of this platform starts from using SSIMP because we needed densely imputed GWAS tables for X number of traits, and we didn't want to find proxies. We wanted the whole GWAS table, so we looked for a tool how to upgrade these old GWAS from HapMap density to the 1000 genomes density or to the HRC density. So in this Manhattan plot A, you see that this is the top locus for HDL cholesterol, one of the top loci for HDL cholesterol from the paper of Kristen Miller from Nature Genetics 2013. So on A is original what she found, and on B is what she would have found if she had used 1000 genome density at the time. Right, so you end up with more significant findings. It's obvious that's also one reason of increasing imputation density to get closer to the causal variance. So we started with putting these tools together, which we would like to use ourselves. So these tools were the summary statistics imputation tool, SSIMP. This is from the group of Pasaniuk, I think, and later on Zoltan Kutalik took it over. Another tool, very important tool, is a liftover. So this updates the positional mapping of the SNPs according to the new genome build you want to use. And then there are tools for generating new data. So this is a tool that is developed by Michel Nivard. I think he's still from Amsterdam, right? And so this is basically, at the moment, this was used for calculating HOMA insulin resistance from insulin and glucose. And there is a paper by, I believe, Irina Fetko. So what this tool can do is that you want to make association statistics for composite measures. Say it is a HOMA, say it is BMI. What you can do, you can use for BMI, you can use height and weight, and you can get the summary statistics of the statistics by putting them in a formula, and then with some inflation or deflation or some approximate estimates. Of course, it is not going to be perfect, but you have an idea of what your BMI, what your effect estimate for BMI would be for this particular SNP. And then there is another tool which is called SumScopa. This is a tool that is being prepared by Marika Kinen from our group, and it performs multi-phenotype GWAS from existing summary statistics data. I believe this is something in line of MTAG. And then there are tools for running multi-phenotype analysis. Multi-phenotype analysis is a focus of our research group, and these are SCOPA for common variants, MAR for rare variants, and METIL-SCOPA for multi-phenotype epigenome-wide association studies. So multi-phenotype methods, basically, I'm sure there are lots of people who don't use it. I also didn't use it until I joined this group. What it basically does, it takes the SNP or the gene methylation as the outcome, and puts the multiple phenotypes, multiple correlated phenotypes as the predictor of this genetic or epigenetic variant. Therefore, by looking at, by including phenotypes with moderate correlation with each other, it boosts the power and therefore the discovery. And then there is also a tool for multi-phenotype imputation, which is ImputSCOPA. Basically, it's a multi-phenotype imputation, but we are implementing several methods in that, which one of them is going to be CRILC, and one of them is going to be the random forest. So we put all this together in this interface of Anglia. It's an interactive interface, a bit like the LD Hub, a bit like the what's it called, MR-based, that you can upload your data, you can generate your library, and you can use the tools from the platform in an online manner without needing to download it and install it in your computer. As I said, what it does, it deals with individual level, summary level, and summary level data, and also there is a library function where we dump the genome-wide association studies, which we already upcycled. So what can user do? They can upload their own data or download from the library and use them in analysis as desired, or they can use the imputation model. They can do the phenotype imputation, or they can direct these imputed phenotypes and they can feed them into the analysis model, which they can run together. Of course, they also need genotypes, which they can run then SCOPA, METIL-SCOPA, and MAR for rare variants, and then everything is shared in the library. And if you want, you can make them, you can share them with public or not. And then there's also this upcycling data model. You put your GWAS summary statistics from HapMap, and then it will upcycle it to the imputation density of HRC. And using the SSIMP. And another thing is the data generation model, which I talked about, is performing multi-phenotype analysis from summary statistics and getting estimates for composite measures as with GWAS. So there's a demo version which is still very preliminary, but we want to make it this up and running very soon so that we can all use it. So that was my conclusion a little bit. I think it's a bit more about strategies to overcome your challenges, but also hugging your challenges, because you won't be able to overcome those challenges because we are limited with the availability and collaboration and manpower. And an acknowledgement slide for referring to people who worked on it. Yeah. So that's it from me. I'm sorry, it was too long.
Thank you very much, Ayşe, for this very comprehensive overview of available data sets and analysis tools for integrating cross-omics data. And also especially for sharing your experiences with some of the challenges that you encountered. I'm going to look if we have any questions, and there is a question from Henk about imputation methods. Can we unmute Henk?
I hope I can answer.
Let me see. Can you hear me?
Yes.
Okay, so you showed that you need imputation to increase your power for all of these analyses that you're doing, if I understand you correctly. And now I wondered, so these imputation, they seem a bit, they seem advanced. So you use other data, other metadata to infer things that are not there yet. So did you do any analysis, like leaving out known measurements and seeing how good these are? And can you sort of share what kind of
Which imputation are you talking about? Are you talking about genotype imputation based on summary statistics, or phenotype imputation?
So you were doing the phenotype the phenotype imputation.
So multi-phenotype imputation. You're talking about multi-phenotype imputation.
So yeah, and any of those. So I'm not completely. So I'm a bit new to this particular type
Sure.
field,
So, well, imputation is something we use very widely, and we have to use it very widely. It's not only for increasing power, it's for making things comparable. For instance, in terms of genotype imputation, I have a data set, I run analysis, and then I ask, for instance, Jenny to check my top SNPs in her data analysis, in her own data. And if we are using different chips, different arrays for genotyping our data, we can't find a common ground to speak, right, with Jenny. So therefore, whatever array we are using, we input our genotypes to the reference panel, whatever best and whatever suitable for the population is. There are different ones for different ethnicities, which perform better on different ethnicities, with different sample sizes, of course, all of them. But this is like a standard. I don't think anybody nowadays uses genotype data as it is to perform analysis. We use it for QCing the genotype data, for excluding outliers, outlying individuals. We use it for estimating relatedness, or for identifying outlying individuals, or for principal component analysis, but we don't use it for association analysis anymore. So this is about the genotype data. It's not only power, it's key to integration. And phenotype imputation is something else. There are different methods, and it really matters a lot depending on the missingness of your data. So there are different types of missingness. There is a random missing, semi-random missingness, and missingness that is totally at random. Mm-hmm. For example, if you want to impute metabolomics data has always has missingness on the left part of the distribution because of values that are not detected, or because of values that are not detected with confidence, which would be below the detection limit, or quantification limit, or semi-quantification limit, whatever it is, right? So we always have NAs. For example, if you want to impute metabolomics data, we actually tried it. A student of mine tried it actually, and it was very interesting to see. If you want to include your metabolomics data with a random forest model, for example, which assumes that your data is at random, mm-hmm. it is missing at random or semi-random. What it does is that I wish I had the figure here easy to show. It's everything that is supposed to be below the imputation, below the detection threshold, is imputed to the mean. Right? Mm-hmm. So this is something that you really don't want. So it depends on the type of the missingness in your data. For instance, for this type of missingness, due to the limit of detection, you should use KRILC, KRLC. So that imputes the extreme values on the left-hand side, and it imputes very well. But you can't use it for the right-hand side because the algorithm assumes that everything is below the lower detection limit, but it doesn't understand if they are higher the upper detection limit. So it's never perfect.
Okay, but it really depends on what you're looking at. That's what you're saying as well, right? So
exactly. Also, what you want to include in multiple imputation, what kind of variables you want to include there. So you can. It is always good to run a couple of test runs and compare the performance. Where does these values end up? Do they end up where you expect them to be, or they end up in the middle of the distribution? So there are multiple solutions.
okay, right. Thank you.
You're welcome.
Thank you for your question. Are there any other questions?
Well, I'm still able to talk, Peter Bram, het Hoen here. So really impressive work, Ayşe. So I have a more general question on multi-omics data integration. That was the topic of your seminar, right? So is it correct that you actually apply this multi-omics integration for two purposes? First purpose is say on the genetic side to zoom in on causal variants or likely causal variants, and the other part is more on the other side, so to get a better understanding of the mechanistic aspects that explain these associations.
Yes, indeed. I think you summarized it very well. So when we have this data from this genetics, transcriptomics, exposome, etc., why we have all this data? We want to have a story, right? We want to understand the story, and we want to be able to put them all on the same picture. And well, that is possible, but it really depends on the. I think the focal point is especially in integrating multi-omics data. The focal point is the availability of GWAS. This is how I see it, because then you can link everything to everything using those tools. And the other part is understanding causality. This is actually totally different. This is because in talking about Mendelian randomization doesn't care which pathway, which genetic variant you are using as instruments. It only looks if they are strong enough. So it doesn't give us any insight as it is, what is the causal path, what is the biological mechanism. It doesn't give insight. It just tests. If I use a random instrument that's not unconfounded, can I predict this phenotype or not? So indeed, if that's what you mean, yes, you summarize it very well.
Well, thank you. That was a good follow-up then. Thank you.
You're welcome.
Thank you. We have a question from Anna.
Yeah, I have, well, first of all, thank you for your really nice talk. I have one question regarding your toolbox or platform Anglia. So it's quite a collection of different tools. And if I were to analyze my data set with different tools, is it possible to share the entire workflow with another researcher so that he could perform the same analysis on their data set?
Yeah, I think that is something at the moment we didn't. When we designed it, we didn't think of that part, but I think it would be very good if we could compile them into like a real, if we put combine them in real workflow engines that packed with all dependencies and could be distributable. I think we could even share it on the website itself. So the website and all platform, all tools there with all dependencies I think this is something we should do, that should be downloadable with a Docker image or something, and then you should be able to install it on your own machine. So like downloadable as a container image, what they call virtualization container or something like this. So I think we don't have it at the moment. We don't have it at the moment. We have all of them separately. So this is pretty much a work in progress. But we want to finish this and yeah, and spread the word so people start using it. So it all started because we needed these tools.
Yeah. Would you then also say that having something like that ready for the data that you integrated, so that there was some analysis already done to this data before you integrated them? If you had this analysis ready in a container or in a workflow, would that have made your research easier?
Oh yeah, of course. Well, for example, if I had this, yeah, it is a problem. For example, it is a problem with, I really like GWAS Atlas, but it also has the same problem. For example, when you look for a SNP, you want to perform FIVAs. For some SNPs, you have FIVAs of 4,000 phenotypes, and for some SNPs, you have FIVAs of only a couple of hundreds of phenotypes. So it's quite annoying, and the reason behind it is that these phenotypes, hundreds of phenotypes, are still in the HapMap imputation panel. So actually, we spoke with people who were developing the GWAS Atlas. If we could update all these we could upcycle all these old association statistics using SSIM, for example. She's going to, or they are going to make a new link, new addition to the GWAS Atlas, and then those SNPs are not going to be missing anymore, and the FIVAS analysis will be complete. Yeah, indeed. Things are improving, right, every year. If we would do the same research this year, it will be different.
Yeah, thank you.
Thank you very much. Are there any other questions for Ayşe? If not, then I would like to thank all the participants very much for joining us today. And Ayşe, of course, thank you very much again for being with us. And I hope to see some of the participants from today also next week in our next webinar on Tuesday.
Yeah, also from my side, thank you very much, Ayşe, and also thank you, Jenny, for sharing the session.
Yeah, thank you very much. It's a great experience. This is my first seminar online, actually. Can you believe? So.
Yes, we'll have more of those in the future. Okay, then thank you, and I hope to see you next week.
Okay, bye-bye.
Thank you.